The ability to clot blood is a remarkable phenomenon that many people probably do not understand in full. Imagine a lifestyle in which your body did not possess the ability to clot blood. In this lifestyle, even the smallest cut could become life-threatening. You most likely would have never learned how to ride a bike, for that would surely be too risky. You would never have been able to play on the Little League baseball team. Although this lifestyle may seem frightening and unrealistic, it is not all that uncommon. In fact, the inability to clot blood is a defining characteristic of the unfortunate victims of the hereditary genetic disorder called hemophilia. Fortunately, our bodies possess the ability to clot blood. Shortly after cutting yourself, your body forms a fibrin mesh composed of platelets that block the flow of blood out of the cut. The molecular pathway leading up to this result involves a number of steps and intermediates. An anticoagulant could act on any of these steps to disrupt blood clotting. Now you may ask yourself, why would we ever want to prevent blood clotting? That is, under what circumstances would preventing blood clotting be favorable? Turns out there are a handful of reasons when such an action would be favorable, and nature itself has found some creative reasons as well. For example, the humorously named protein called draculin is used by vampire bats to prevent clotting of the animal's blood that it is drinking. More relevant to humans, there are a variety of clotting disorders, such as thrombosis and pulmonary embolism, or more familiar ones like stroke and myocardial infarction, also called heart attack, that could be treated with anticoagulants.
The compound we have chosen for our project, warfarin, works by inhibiting vitamin K epoxide reductase, VCOR, an enzyme responsible for the reduction of vitamin K. Presence of reduced vitamin K is necessary for the activation and proper function of a second enzyme called vitamin K-dependent gamma carboxylase. This enzyme carboxylates glutamate residues to gamma carboxylglutamate residues specifically on prothrombin and coagulation factors 7, 9, and 10. The newly carboxylated gamma carboxyl glutamate residues of these proteins are able to readily bind calcium ions, ultimately forming the fibrin mesh and platelets that cause clotting. Thus, if warfarin inhibits VCOR, the pathway cannot proceed.
Here is the protein vitamin K epoxide reductase. The active site is located within the 4-helix bundle shown in pink and contains the structure ubiquinone and two nearby cysteine residues. Attached to this 4-helix bundle is the TRX-like domain, or thioredoxin-like domain. The introduction of vitamin K into the active site of VCOR initiates a pathway in which the vitamin K epoxide is reduced to reduced vitamin K by a series of redox reactions involving CXXC motifs, which are two cysteines with two other residues between them. The sulfurs of these CXXC motifs are reduced from disulfide bridges into two thiols. In a series of steps, a yet unknown protein reduces the CXXC motif in the TRX-like domain, which in turn, true to its name, thioredoxin, reduces two cysteines located in the half loop of VCOR, forming two thiols. The reduced cysteines within VCOR react with the cysteines located in the active site, reducing them to form two thiols. Here you see them as a disulfide bridge. Once these thiols are formed, the CXXC motif in the active site reduces the vitamin K and regenerates the disulfide bridge, and vitamin K is reduced.
Shown here is the vitamin K-dependent carboxylase reaction. Reduced vitamin K is used as a cofactor for this enzyme using oxygen, which adds here, and carbon dioxide, which adds here. In a beta elimination-like reaction, an epoxide as well as an oxyanion is formed. This anion then deprotonates the syn hydrogen on glutamate, generating a carbanion. In the following step, the carbanion attacks an incoming carbon dioxide molecule, leading to the formation of the product, gamma carboxyglutamate residues. The remaining vitamin K23 epoxide is recycled back to reduced vitamin K in a two-step process catalyzed by VCOR as discussed previously. Carboxylation of these residues, located on prothrombin and coagulation factors 7, 9, and 10, activates them, leading to the interaction with fibrin, leading to blood clots. When warfarin is introduced into the bloodstream, it inhibits VCOR, thus preventing the clotting process. Although this process is still unknown, we do know that either vitamin K or warfarin will fit into the active site, but not both. Furthermore, mutations in residues which are implicated in binding vitamin K, shown here in blue, result in the resistance of warfarin, suggesting that warfarin binds the active site of VCOR. Therefore, when warfarin is present in your bloodstream, VCOR is inhibited, and so there is no reduced vitamin K for vitamin K-dependent gamma carboxylase, which, as noted earlier, activates the precursors to blood clotting agents. Thus, you are blood clot free.
Overdose of warfarin can result in massive hemorrhaging, which can be lethal. Indeed, for this reason, warfarin is also used as a rat poison. Further, Russian historian Vladimir P. Naumov and Yale University Soviet scholar Jonathan Brent suggested in a 2003 book that warfarin was used to murder Joseph Stalin. Finally, we would like to leave you all with the real-life application of warfarin, also known as Coumadin.